Biotechnology, figure 11.4. So E. coli cloning vector PBR 322 we are discussing. In order to link the alien DNA and the foreign DNA of interest, the vector needs to have very few, preferably single recognition site. For one restriction enzyme, presence of more than one recognition sites within the vector will generate several fragments, which will complicate the gene cloning process. So if you want to flank the DNA at this particular region, so there should be one cut site or one cut site. So multiple cut sites will result in complication. Next, the ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. ಇಲ್ಲಿ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಇದೆ, ಇಲ್ಲಿ ಕ್ಲೀವೇಜ್ ಸೈಟೂ ಇದೆ, ಇಲ್ಲಿ ಬ್ಯಾಮ್ H1 ಇದೆ, ಇಲ್ಲಿ PBUಗೆ ಇದೆ, ಇಲ್ಲಿ PSTಗೆ ಇದೆ. ಸೊ ರೆಕಗ್ನಿಷನ್ ಸೈಟ್ ಇದೆ, ಆಮೇಲೆ ಮಾರ್ಕರು ಇದೆ, ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸು ಇದೆ. Suppose if we cut this region and we insert the foreign DNA, so the tetracycline resistant gene may not be functioning. This particular vector loses this capability of resistance against the antibiotic tetracycline. This is known as insertional inactivation. Due to the insertion of a gene, alien gene, the property of this gene will be lost. Activity will be lost. Is known as insertional inactivity. So this is help in selection of recombinants. Here we take out this plasmid and we treat with the restriction endonucleases and we ligate the gene of interest. So if the ligation is successful, the recombinant plasmid will be formed. If there is no ligation, non-recombinant, this becomes non-recombinant plasmid. Okay. So the recombinant plasmid has to be uptaken by the host cell. So how to incorporate this vector recombinant plasmid into the host? By treating with the calcium ions, divalent ions, and heat shock. So incubate in the ice, suddenly raise the temperature at 42 degrees centigrade, that is a heat shock, and then again incubate in ice. So the disruption of the membrane results in the uptake of this foreign DNA, vector, and that results in the transformant. So if it is transformant E. coli, when it is grown in, say for example, it is having the ampicillin resistant gene and tetracycline gene get insertion inactivated, so only one gene is functional. So if we incubate this particular transformant in the ampicillin, in the medium containing antibiotic ampicillin, this will grow. The transformant will survive and we can select this transformant. If the non-transformed E. coli will also perish because it don't have any either of resistant gene. So here in this case, one problem may arise. If the non-recombinant plasmid arises, what happens? It grows both in the antibiotic, ampicillin resistant, and also in the tetracycline resistant. To overcome this problem, we are trying to introduce the alien DNA between the LAC-Z. This LAC-Z gene will produce the enzyme beta-galactosidase. This enzyme will convert that chromogenic substance like X-gal, 5-bromo-fluorochloro, 3-indole beta-galactopyrenosidase. It's a chromogenic substance that if the chromogenic substance present in the medium, if the plasmid containing LAC-Z gene, it produces the beta-galactosidase. This beta-galactosidase will convert this X-gal into the blue color, dark blue color. This dark blue color indicates the non-transformants. The white colored colonies, non-blue white colored colonies, is the transformant. Why? Because so we have the insertion site between in the middle of the LAC gene. So the introduction of the alien DNA in between this LAC gene will result in insertional inactivation of this LAC gene. So beta-galactosidase won't be produced here. So the colony will be white. So this is a very clear-cut differentiation where we can isolate these distinct colorless colonies as a transformant colonies. This blue colonies are the blue colored bacterial colonies are the non-transformant, contains the non-recombinant plasmid. Okay. Inactivation of the gene for synthesis of this enzyme is insertional inactivation. Vectors for cloning genes in plants and animals. So, the Agrobacterium tumeficans, a pathogen of several dicot animals, is able to deliver a piece of DNA known as tDNA to transform normal plant cells into a tumor cells because it has a Ti plasmid is there. The retrovirus in animals have ability to transform normal cells into cancerous cells. So we will use the mechanism of the transfer of gene from these organism into the higher plants or higher animals apart from the bacteria. The tumor-inducing Ti plasmid of Agrobacterium tumeficans is the cloning vector where it is not pathogenic, but we can use the mechanism to deliver genes of our interest into a variety of plants. So in the same way, retroviruses have been used to disarmed and are now used to deliver desirable genes into animal cells. Next is another way of introduction of gene is the microinjection method. Recombinant DNA is directly injected into the nucleus of an animal cell. For plants, because they have the cell wall, the cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA is a method known as biolistics or gene gun method. And the last method uses disarmed pathogen vectors, which when allowed to infect the cell, transfer the recombinant DNA to the host. Processes of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction into nucleases, isolation of desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at large scale, and extraction of the desired product. Isolation of genetic material. So genetic material can be isolated of interest. So they can be isolated from bacteria, plant, fungus, whatever it is. So like a lysosome to dissolve the bacterial cell wall, cellulase, plant cells, chitinase, the fungal cell wall. The fungal cell wall is made up of chitin. So the genes are located on long molecules of DNA, intertwined with proteins such as histones. The RNA can be removed by treatment with a ribonuclease, whereas proteins can be removed by treatment with proteases. Okay, so as you recall the Avery experiment, so you would have knowledge in the getting of purified DNA, and the purified DNA ultimately precipitate out after the addition of chilled ethanol. This can be seen in the diagram. You can see here when the chilled ethanol is added to this particular extract in solution, it will be precipitated. You can see this.